Welcome to all of you dear students. In our today's lecture, we will be discussing the process of digestion in our stomach. So far as the different anatomical structures and histological aspect of our stomach and other parts of the digestive tract are concerned, you can go through my previous lecture which is available on my YouTube channel. And here in our today's lecture, we will be having a thorough look over the process of the gastric phase of our digestion. Before having a mechanical look over the mechanics of the process of digestion in our stomach, let us have a look over the typification of the different glands which are present in our stomach. You know, our stomach has been provided with different glands or different secretory cells which have been divided into three types. The first type of our stomach gland is known as cardiac glands. In our stomach we have cardiac glands, and most importantly we have second format of the glands in our stomach, and that are called as the funding glands, and then we have third format of the glands lying along the lining of the wall of our this stomach, and those glands are called as pyloric glands. Let us have a brief description over all these three types of glands which are present in our stomach. So far as the cardiac glands of our stomach are concerned, they are very small. And they are of the tubular form and racemose form. They are very small and they are of tubulo-racemose type. Very simple to remember that cardiac glands are small and they are of tubulo-racemose type. So far as the cells present in this very type of the gland is concerned, they mostly consist of columnar cells. And all of you know that columnar cells are responsible for the synthesis of mucus. For mucus secretion or mucin synthesis, columnar cells are responsible. And then we have second type of the gland, that is the funding glands, and funding glands are of compound tubular type. They are compound as well as tubular. So comparatively they are larger and they are of compound tubular type. And these types of the glands in our stomach have different forms of the cells responsible for synthesis of different products. Number one and important cell which is present in the funding gland is called as chief cell, which is a polyhedral cell, and this very chief cell is also known as peptic or zymogen cell. So I will write down here, it is also known as peptic or zymogen, isn't it? And you should remember here one important thing, that this chief cell or the peptic cell or the zymogen cell of the funding gland of your stomach is responsible for the synthesis of so many things. Like it is responsible for the synthesis of an important inactive format of an enzyme that is known as pepsinogen. It can synthesize pepsinogen, the function of which will be discussed in the later part. And these peptic cells are also responsible in some stage of our lives to synthesize another inactive format of another enzyme that is known as prorenin. This is not that renin which is being synthesized from juxtaglomerular apparatus of our nephrons in our kidneys. No, in that renin, when we spell that, we write down there REN, single N, then IN, And so far as this very renin is concerned, we write down here RENNIN. This renin. This is with which you can differentiate that nephric renin with the gastric renin, isn't it? And cheap cells or peptic or the zymogen cells are also responsible for synthesis of another important enzyme with which some sort of lipid digestion takes place in our stomach, and that very enzyme is called as gastric lipase. Gastric lipase. So, besides the presence of chief peptic or the zymogen cells in the fundic glands of our stomach, we have also some other types of the cells which are present in these fundic glands and which are responsible for the synthesis of a very important gastric secretion known as hydrochloric acid, isn't it? The most important feature that is lying or that is associated with the gastric part of our digestion, that is the synthesis of highly acidic substance, that is hydrochloric acid, by our stomach. And the cells which are responsible for the synthesis of that hydrochloric acid, they are called as Oxentic. They are called as Oxentic cells, or these Oxentic cells are also called as parietal cells. These are also called as parietal cells, and it is these parietal cells which are also responsible for the synthesis of some other important secretions in our whole of the gastric secretion. One of the important secretion here is HCl, this acid with which so many activities with respect to the digestive process takes place in our stomach. So HCl is being synthesized by the Oxentic cell of our fundic glands or by the parietal cell of our fundic glands. Besides the ability of these Oxentic or the parietal cells for the synthesis of the secretion of the HCl, they have also another ability for the synthesis of some factors, some chemicals which are known as intrinsic factors. They also synthesize, I will write down here IFs. Here IF refers to the intrinsic factors. Actually, they are named as Castle's intrinsic factors. They have their own function about which we will come to know in the later part of our lecture. So, this is the second format of the cells which are present in our fundic glands. Then let us have a look over the pyloric glands which are also present in most of the pyloric region of our stomach. And in these pyloric glands, we mostly have these columnar cells. Besides having the prevalence of the columnar cells in this region, we also have another type of the cells which are called as G cells and another type of the cells which are called as D cells. Another cell G cells and another type of cell, that is D cells, isn't it? And so far as the secretion of the columnar cells is concerned, they are mostly responsible for the synthesis of mucus, the function of which will be discussed in the later part. And both these cells, G cells and D cells, are responsible for the synthesis of hormone-like substances. Like gastrin is there for the synthesis of hormone-like substances which indirectly regulate the gastric secretion or the function being done by our stomach. So far as the pyloric glands are concerned, so far as their typification is concerned, they are of the compound tubulo-alveolar type. They are compound, they have tubulus, and they have alveolar type of the tubulus. So they are also called as, we have to put them into the category of compound tubulo-alveolar type. Now let us have a look what the brief description of the different types of the glands which are present in our stomach. We have seen we have cardiac glands, we have fundic glands, and we have these pyloric glands. And in our cardiac glands, so far as their typification is concerned, they are very small and they are of the tubulo-racemose type, and their main cells which are present in them are these columnar cells which are responsible for the synthesis of the mucus. Then we have fundic glands which are of the compound tubular type, and their primary cells are chief peptic or these zymogen cells which are responsible for the synthesis of pepsinogen, prorenin or gastric lipase. Then we have another type of the cell which is present mostly in the fundic glands. They may be present sometimes in the pyloric region as well, but mostly they are present here in the fundic glands, and which are called as Oxentic cells or the parietal cells, which are responsible for the synthesis of two important things. One is hydrochloric acid and another is gaseous intrinsic factor. And third type of the gland is pyloric gland, which is of compound tubulo-alveolar type, and main cells present in this very gland are columnar cells for the synthesis of mucus, G cells and the D cells for the synthesis of different hormones. After having a brief description over the different types of the glands which are present here, now let us have a look over the mechanics or the nervous mechanics or the nervous and endocrine mechanics which are collectively operating over the lining of the stomach, so that the gastric juice is synthesized at the time of the need and the gastric digestion takes place in an efficient manner. In order to understand well the mechanics operated over the stomach, we have to focus on first the reflex arc associated with the whole of the process of the digestion taking place in our stomach. So, the whole reflex or the nervous control which is being operated over the stomach wall can be divided into three phases. Number one, of which is called as cephalic phase. Very simple to remember. Number one is cephalic phase. Number second and most important one is gastric phase, and number third one is referred as intestinal phase. What we are saying? We are saying that so far as the mechanics of the nervous control of our gastric secretion is concerned, we have divided all that control into three important phases. Number one is cephalic, number second is gastric, and number third is intestinal. Let me tell you that so far as the cephalic phase of our nervous control of our gastric secretion is concerned, this phase takes place before we have incorporated the bolus into our stomach. This phase actually this phase acts as a preparatory phase for the process of digestion which has to take place within stomach. So, in the cephalic phase, there are certain things which are responsible for the induction of the gastroenzymatic secretion or for the secretion of the gastric juice, and the different parameters which are responsible for positive effect in the cephalic phase, which are responsible for the secretion of the gastric enzymes or the gastric juice, they include a good smell. When you smell an eatable, they include the sight when you see, when you are having a look towards an eatable, then you get induced or your stomach gets induced for the synthesis of the gastric juice. And not only that, even when you think or different thoughts, they can also be associated in the cephalic phase for the synthesis of the gastric juice. And besides that, sometimes the taste as well. When you have just incorporated something into your eatable into your buccal cavity and you have tasted it, your tongue has tasted it well and you have felt well, then that good taste will also induce in your cephalic phase of the gastric secretion for the over secretion of the gastric juices. But this all accounts as a preparatory phase for the process of digestion, which has to take place in what? In your stomach. And there are some also negative factors which can inhibit all these things, isn't it? They have induced, they are as the inductors for the sort of your cephalic phase, but there can be some other factors like, let me write down here some other factors like citrus is there, like emotional distress is there, like some sort of heartbreaking. Let me write down here. By writing down here heartbreaking, I mean to say some emotional conditions in which we feel very bad and we are not mentally very sound at that time. Such thoughts, such emotional distress acts as an inhibitory thing for the synthesis of the gastric secretion, isn't it? And besides that, there are some other factors as well which are also responsible for cessation or the stoppage of the gastric secretion. That may include some drugs, that may include some medicines about which we will come to know in later lectures. But in the cephalic phase, there is only synthesis of the gastric enzymes in such a way so that our stomach prepares well that now bolus is coming or bolus is about to come into my lumen and I have to digest it. So the preparatory phase is accounted in cephalic phase of the mechanics of gastric secretion. Then there comes the next phase, that is the gastric phase, in which we will come to know that how actually the process of digestion takes place under the influence of these glands or the cells which are present in these glands, and what are the factors which are responsible for the synthesis or the secretion of these substances at that time when we need them, when they are necessary, isn't it? So the second phase in our mechanics of the gastric secretion is called as gastric phase. And in gastric phase, what happens? In gastric phase, it is the food which has been just incorporated into the lumen, then this food is accounted and then this food gets accumulated as if in the form of the concentric circles. The food which has been just incorporated into the stomach will be here, and the food which has been incorporated previously in the stomach, that will be very close to the wall of the stomach. And as soon as the food reaches and it actually attaches towards the wall of the stomach, that may lead to distension of the stomach wall. That may lead to distension of stomach wall, isn't it? Once the distension of the stomach wall takes place, then we will see that how different neurons which are present in the submucosal layer of our stomach wall, how they induce our vagus nerve or our nervous system to initiate these digestive glands for the synthesis of gastric secretion. So this distension can be an inducing factor. Number second, the hormones like gastrin, we have seen that the hormones can be synthesized by such type of the G cells. That is why G cells are called as so, because they are responsible for the synthesis of what? For the synthesis of the gastrin hormone. And it is this gastrin hormone which will also be responsible for the induction of different types of the cells present in different types of the gastric glands to initiate them for the synthesis of their respective secretions. And besides that, there can be so many other factors which induce our nervous system first, then the nerve centers, then the reflex centers, then the vagus nerve, then ultimately our different glands or the cells which are present with the lining of our stomach to induce them for the synthesis of different gastric secretions, isn't it? Then in the third phase, we will come to know in the intestinal phase or the third phase, we will come to know how the regulation of this gastric secretion takes place. In the intestinal phase, we will come to know about the regulatory mechanism, regulation of synthesis of gastric juice or gastric secretion, for which different factors are responsible, like somatostatin is there, like gastric polypeptide is there, isn't it? Like decrease in chyme content is there, decrease in chyme content, and some psychological factors are also there. Some psychological factors are also there responsible for the intestinal phase or regulatory phase of the gastric secretion. Besides that, so far as the regulatory phase of the gastric secretion is concerned, we have seen that there are some other factors as well which are responsible for the same, like that of VIP or vasoinactive polypeptide is there, and some other types of the intestinal polypeptides are there. All these factors will be collectively responsible for having a regulatory control over the secretion of the gastric, these cells which are present in different formations of the gastric glands of our stomach. So now let us have a look over the mechanics being operated in different phases of the gastric secretion. For that, let us have a look over this very diagram which I have made at an earliest. See, if this is the cerebral cortex, if this is the brain of a person, and let me write down here, this is the cerebral cortex, and if in our brain, with the help of all these things which are responsible for this indirect synthesis of the gastric secretion, then all things will lead to induction of the cerebral cortex to induce or to send signals to the next region of our brain, that is hypothalamus, to send indirectly a message to the respective center for the gastric secretion, which is located in medulla. In the medulla, there is a specific gastric secretion controlling nucleus which works under the influence of the hypothalamus, which works under the influence of what? Cerebral cortex. So cerebral cortex has guided hypothalamus, and as a result of which it has guided what? Your medulla. Then your medulla will do one thing, that it will now copy the message and will send it to the lining of the stomach wall via this very nerve, which is known as vagus nerve. This is vagus nerve. In your previous lecture, you came to know that there is not only this vagus nerve which is responsible for the induction or regulation of the gastric secretion or the digestive processes. You have seen that another nerves like the trigeminal nerve is there, which is located towards the backside of our facial region. And there is glossopharyngeal nerve, isn't it? There was 12th cranial nerve as well, which were collectively responsible for the deglutition process and which were collectively responsible for the happenings of the digestive tract. Similarly, such other nerves are also responsible for having a little bit regulation over the gastric secretions. But the main mechanism here is that once these things induce your cerebral cortex, then it directs your hypothalamus, that indirectly directs your medulla oblongata, and through the vagus nerve it sends messages to your stomach wall for having their respective synthesis of the respective cells of the respective different types of the gastric glands. Besides that, so far as the anatomy of our gastrointestinal tract is concerned, I already told you that we have Meissner's plexus is there, plexus of Auerbach is there, isn't it, or myoenteric plexus is. Let me write down here that here as it is in the submucosal region of our gastric lining of our stomach, it is myoenteric center, myoenteric reflexes, myoenteric plexuses have specific nucleated centers, and they also have the ability to send internal messages to the respective part of your central nervous system for having an regulatory and arced control over the synthesis or secretion of the gastric juice, isn't it? And it will send signals from, and these signals will come towards the stomach. Besides that, in the lower part of our medulla, we have some sympathetic centers as well. Here we have sympathetic centers as well, which are sending the inhibitory signals. They are sending the they are sending the inhibitory signals towards the stomach wall. The inhibitory signals which work against the initiating signals of the vagus nerve. So, this is how the regulatory arched control operates over the lining or over the different digestive cells or digestive glandular cells of our stomach wall. And here let me write down as if here it is the sympathetic nucleated control, and it will be having a regulatory control, and this will be having the initiating control. These are the primary nervous mechanisms operating over the wall of the stomach for having the gastric part of our digestion. Now, as you know, so far as our previous lecture is concerned, I told you that our stomach has a greater curvature, it has a lesser curvature, and it has been divided into different regions. The first region, say here, it was called as cardiac region, and the next region, say here, it is known as fundic region, and the next region, say here, this was known as body, and the next region, pyloric antrum and pyloric canal, isn't it? But in order to make us understand well how the mechanism of synthesis of hydrochloric acid, how the mechanism of synthesis of pepsinogen, how the synthesis of mucin-like substance takes place along the lining of our stomach wall, I have made some only a few cells here, isn't it? Only a few cells here, and for that, let me write down here as if this is the parietal cell. This is the parietal cell here. Similarly, I have made another cell, the another one, that is this peptic cell. Here it is peptic cell, and the third one, say this is the G and D cell. This is the G cell, and this is the D cell, isn't it? Besides that, I will make some smaller, simpler cells which are called as columnar cells. So these are the columnar cells. This is not the histological perspective. This is only a diagram with which I can make you easily understand the things which are happening in the digestive process. This is not the real histological diagram. This is only the diagram which I have made to make you understand the things or the happenings in our stomach for the process of digestion. So, the first thing is that what is the mechanism which is being operated by the parietal cell for the synthesis of an important secretion, that is hydrochloric acid. All of you know that this parietal cell of this funding gland has tremendous amount of mitochondria. Enough mitochondria is there. When more and more mitochondria, why mitochondria is more there? You know that mitochondria is in more quantity in those cells which are mostly active, like our muscle cells. Our muscle cells are syncytial because more mitochondria has to be there, because more oxygenation process has to be there, more glycolysis has to be there, more consumption of the oxygen has to be there. Wherever the consumption of the oxygen is to be more and more, there it is necessary for that cell to have more and more these energy factories, ATP factories, that is the mitochondria. When this parietal cell has enough mitochondria, then definitely more this glycolysis process will take place. When more glycolysis process will take place, then definitely there will be more and more release of carbon dioxide. When more release of carbon dioxide will take place, then the pH conditions, acidic or the basic conditions of in the cytoplasm of this parietal cell will definitely not remain as the condition will be prevailing in other types of the cells of the body. And when the prevalence of the more carbon dioxide will be there, then it will be mandatory for this very cell to operate a mechanism to have a regulatory control over this oversynthesis of what? Carbon dioxide. When more carbon dioxide is there, then this cell has to make use of its water. See here. Now it will make use of its water and it will ultimately actually divide this water into H positive and OH negative. How? Via this reaction, it will convert these two products into H2CO3, and finally this carbonate will be converted into H positive plus HCO3, carbonic ion will be there negative, and this reaction will take place under the influence of the enzyme known as carbonic anhydrase. Let us suppose that this is the enzyme. I will write down here Ca, this is the carbonic anhydrase. So, carbonic anhydrase of the parietal cell has been used to manage the high concentration of the carbon dioxide by mixing it with water in such a way so that it will be converted at CO3 first, then it will be converted into the H positive protons and these carbonic ions. And these H positive ions will be transported via specific channels towards the outside or the lumen. They will be transported towards the lumen or canaliculi of the duct. You know, the funding glands in which these cells are present, they have isthmus, they have canaliculi, they have the bases, and here I have made only this very particular parietal cell. And you have seen that in the parietal cell what the role of the carbonic anhydrase has been here, with which the H positive ions have been synthesized or produced, and they have been secreted out into the lumen of the glands. And what we need the acidic condition in the lumen, then the acidic condition in what? In the whole of the lumen of our stomach, and then that increased hydrogen ion or the protonic concentration in the respective regions will lead to decrease in the pH and higher acidic conditions. But here you can ask me a question that once the H positive ions will get out through the respective channels from the parietal cells, then the imbalanced acidic basic conditions will take place within the cytoplasm as well as outside the cytoplasm or outside the parietal cell or within the lumen. In order to maintain acidic and basic balance, the bufferic agents are being used, the bufferic agents which are present in the blood. Say for example, here I will make a blood vessel here. So, it is the blood vessel or the vein which actually is associated with the lining of the stomach wall, and so far as the blood in this very vein is concerned, it consists of many minerals and many salts like sodium chloride is there, like potassium chloride is there, like many other substances are there about which you already know. And now this sodium chloride will be used in such a way so that the sodium and the potassium will be managed to get transported in this manner here from the extracellular fluid or from the blood. The potassium will come towards the interior side like this. It is the potassium, and usually 2 potassium ions will come and usually 3 sodium ions will go out. Now, this potassium and this sodium will, or their ionic form will be used in such a way so that the potassium will get mixed up interior contents of the cell and it will be mixed with the exterior contents of the cell. And now the chlorine which has been synthesized as such due to the breakdown of the sodium chloride or the chloride which was present or synthesized here by the parietal cell, that chloride which has been formed as such now from the NaCl, now that will be allowed to get mixed with the hydrogen, and when these two will combine, they will result in the form of this strong acid, that is hydrochloric acid, isn't it? So, for the synthesis of the hydrochloric acid, the protons have been produced because of this pathway under the influence of the carbonic anhydrase, and the chlorine which was required for that has been taken out from the extracellular fluid or the region, or the chlorine ions which were present in primarily here in the cytoplasm of the parietal cells. Then that chloride has been shifted out along with the hydrogen ion, and the chloride ion which we got from the extracellular fluid or the blood, all that chloride has been allowed to get mixed with the hydrogen ion in order to form the concentrated format of the hydrochloric acid by the parietal cells. But when this hydrochloric acid has been synthesized, then so many other questions come into play in our brain, that when the 3 sodium ions have been transported out and 2 potassium ions have been transported in, and when the chloride shift has taken place and when the protons have been drifted outside this cytoplasmic region of our parietal cell, then at that time what are the other factors which are acting as buffering agents for having an acidic or the basic balance for within or outside the parietal cell? This ion has to be shifted towards this region in order to maintain the balance against the chloride ions. Since we know that we got chlorine, water, potassium from outside, in order to maintain the balance against the chlorine, we will give this HCO3 to the extracellular fluid or the blood which is present in the respective blood vessel. 1 minute, let me use this color here. We will shift this HCO3 negative ion towards the extracellular fluid region, HCO3, isn't it? Instead of chlorine which has been used here, this will manage the balance, acid base balance, and then this HCO3 here under the influence of carbonic anhydrase will be again converted either into the sodium bicarbonates or potassium bicarbonates. You know that most of the or more than 90% of the carbon dioxide is being transported in the form of the bicarbonates, especially sodium bicarbonates and then the potassium bicarbonates. The sodium and the potassium which is present in the ionic form here, they will be used now by the carbonic anhydrase to convert this HCO3 into the water or sodium bicarbonate or the potassium bicarbonate. Then that carbon dioxide will be transported to for the respiratory processes, which will be discussed in the respiration mechanism. But here we have seen that this carbon dioxide, which has been produced more and more because of overprevalence of the glycolytic apparatus in the parietal cell cytoplasm. This has been used under the influence of the carbonic anhydrase to synthesize H positive and HCO3 negative ions, and these H positive ions or the proton ions have been pumped via the proton pump towards the lumen, and to manage the balance. For that, the shifting of the potassium and the sodium also took place at the very same time, and almost 2 potassium ions were transported at a time towards the inside and 3 potassium were transported outside at the same time, and the potassium and the sodium pump which operated here managed the acid base balance. Besides that, when we incorporated the hydrogen ions outside the cytoplasmic structure of the parietal cells, we let the breakdown of the sodium chloride and we let the chloride ions from the extracellular fluid or the blood to get their entry into up to the lumen or towards the respective gastric cell or the gastric glands, and then that chloride has been used to get fused with the hydrogen to result the hydrochloride acid synthesis. In this way, when the hydrochloride acid synthesis takes place, at the very same time, this potassium inflow and the proton outflow, this potassium inflow and proton outflow, all this is being regulated by so many other factors which we will discuss right now. At that very time, this sodium, this potassium, when the potassium rushes in, this rushing of the potassium in and rushing of the hydrogen out, where a pump is known as hydrogen potassium ionic pump, and until this pump is there, the acid base balance is there and the continuous synthesis and the secretion of the protons are there, which ultimately lead to formation of the hydrochloric acid, or which ultimately lead to decrease in the pH or increased proton ionic concentrations. And now let us have a look over this G cell. This G cell is also known as argentific cell because it is responsible for the synthesis of an hormone known as gastrin. It synthesizes a hormone known as gastrin, which travels via blood. When it travels via blood, and this parietal cell has some specific receptors for this G cell, for this gastrin, it has some specific receptors, and after this hormone reaches via blood up to this very region, then this hormone comes, gets associated with the receptor, and this gastrin then induces the cytoplasm to release, under the influence or under the presence of more and more calcium, to have a good operation over this hydrogen ion pump. So this gastrin hormone is responsible for what? It is responsible for a regulatory mechanism. So under the influence or in presence of the calcium, this gastrin, which has been synthesized here, it will induce the parietal cells to operate all this mechanism, thereby to release hydrochloric acid. And here we have seen that this gastrin has resulted in the synthesis of this G cell has synthesized the gastrin, and this gastrin has induced this cell to synthesize this. At the very same time, this gastrin also induces the another cell, which is the peptic cell, to synthesize an important enzyme in its inactive form known as pepsinogen, Once pepsinogen is synthesized, it comes here out into the lumen. This pepsinogen has come out in a similar mechanical operation through which the parietal cell has been stimulated. I want to say here that the different factors which were responsible for the stimulation of the parietal cell, the same factors are responsible for the stimulation of your peptic cell, for the synthesis of pepsinogen. And now this pepsinogen will come out here into the lumen, and this pepsinogen will be quickly converted under the influence of presence of this HCl into pepsin. And what this pepsin will do? This pepsin will convert. This pepsin has the ability to break the peptide bond, and you know peptide bonds are present in between the amino acids in the polypeptide chains of the proteins. If you have taken a protein rich diet, then this mechanism has been operated by your stomach, and the pepsinogen has been released and it has been converted into the pepsin, and this pepsin has a lead to the transformation of proteins into peptides, into oligopeptides, into monopeptides, due to what? Due to the breakage of peptide bond. It has done the breakage of peptide bond. And remember this very carefully, that once the pepsin has been synthesized, as soon as it has been incorporated into the lumen of its respective gland, it was in its inactive form, that is the pepsinogen. But synthesis of the pepsinogen has been induced by all such factors which have induced the synthesis or the formation of the hydrochloric acid by the oxentic cells. So now let us have a look or the other cells which are responsible for the synthesis of some other factors. Besides that, let me tell you one more thing here, that once these peptides or the protein substances are synthesized, there are some chemical receptors which are present lying over the interior surface of the D cells or interior surface of especially the G cells, and then these respective chemoreceptors get stimulated, and after their stimulation, they once again induce their parent cell for the oversynthesis of their gastric hormone, thereby to induce this and this cell for the synthesis of HCl as well as pepsinogen. Besides that, we also know that the oxentic cell is also responsible for the synthesis of Cassell's intrinsic factor. It also synthesizes another factor, that is, let me write down it here, Cassell's intrinsic factor. And it is this Cassell's intrinsic factor which has almost no role here in the gastric phase of the digestion. This Cassell's intrinsic factor, or it has some other formats of the factors like that of this intrinsic factor. Sometimes they are called as IF1, IF2, IF3, and these intrinsic factors are responsible in the ileal part of our small intestine for the good absorption of vitamin B12. Sometimes there is a type of the anemia known as the pernicious anemia, in which there is deficiency of the vitamin B12, and the deficiency of the vitamin B12 leads to the deformity formation of the red blood corpuscles or the hemoglobin, and at that very time it can be because of under secretion of the Cassell's intrinsic factor with which pernicious anemia is there. So vitamin B12 deficiency can not only be because of the deficiency of the same vitamin in our food, but it can take place because of under secretion of the Cassell's intrinsic factor by our parietal cells. Number one. Number two, if some type of the gastric ulceration is there lying prevailing in most part of our stomach, then that can also lead to under secretion or negligible secretion of the Cassell's intrinsic factor, that ultimately can also lead to that type of the anemia in such patients, isn't it? And besides that, the cell which was responsible for the synthesis of the pepsinogen, it is also responsible for the synthesis of another enzyme known as gastric lipase. We should not rule out that here, that it is also responsible for the synthesis of gastric lipase. different substances, especially the substances which are present in our food rich in lipids or the fat content. When our food is rich in our lipid or the fat content, then it takes enough time for that food to get released out from the luminal part of our stomach. At that time, that timely blockage of that food is also responsible for the induction of this peptic cell for the synthesis of another enzyme known as gastric lipase, which now will be responsible for conversion of some lipids into their minor forms, like glycerol is there, like long fatty acid chains are there. It is responsible for the breakup of the breakdown of the ester bond which is present in our lipids. So partial digestion of lipids can also take place in our stomach when lipids are present in our food which we have taken. Besides that, we have also mentioned here that there are some of our cells which are also responsible for the prorenin synthesis, and those cells are these very cells which were responsible for the synthesis of these two important enzymes. The third important thing which can be synthesized by them is prorenin. But remember here one thing, that prorenin is not being synthesized by we adult people, but it has been seen that it is synthesized by the calf stomach mostly, and it is said in some books that it is also synthesized by the humans in their earlier stages, but in the later stages it has not been found there, isn't it? So this prorenin is or can also be synthesized by this peptic cell under the same mechanism through which other secretions have been secreted by the respective cell, and this prorenin will be responsible for the digestion of casein of our milk, for the digestion of casein to get its respective monopeptide polypeptide chains. So in our stomach we have seen that under the influence of the pepsin, partial digestion of the proteins take place, isn't it? And we have also seen that the hydrochloric acid which has been synthesized by different types of the cells of our gastric glands, hydrochloric acid lead to the transformation of the inactive form of the pepsinogen into the pepsin. Besides that, this hydrochloric acid maintains a pH of around 1.6 to 3.5. It is this range of pH in which the pepsin has been seen and found to act effectively for the degradation of the proteins into its peptides, monopeptides and oligopeptides or even amino acids. Once the pH is raised above than that of the 3.5, then this pepsin has been seen to getting inactivated, then its activity rate reaches at least up to the 0. And one more thing which is associated with the mechanics of the process of digestion taking place in our stomach, that is the role of these D cells which I have made here. D cells are responsible for the synthesis of another substance known as somatostatin. It is a hormone-like substance which is incorporated into the blood, which has an inhibitory effect over the G cells, isn't it? When it has inhibitory effect over the G cell, then G cell will synthesize least amount of the gastrin. When least amount of the gastrin will take place, then inhibitory effect will be well marked by the parietal cell as well as what? By our peptic cell. So least secretion of the HCl will take place from here, and least secretion of these types of the enzymes will take place. So it means that these cells are responsible for having a regulatory mechanics or the secretion of the different types of the digestive glands. And besides that, you can ask me an important question, that if our stomach has the ability to synthesize hydrochloric acid, to synthesize pepsin, why our stomach wall is not itself being digested? This is the question which is oftenly being asked by among the peer groups, by your friends, by in the different examinations. And for that, let me tell you that we have another type of the cells which are called as the columnar cells lying across the wall of our gastric region of our stomach, and those cells are called as columnar cells or goblet cells. It is these columnar cells which are responsible for the synthesis of so many things, especially water, phosphates are there, and an important thing, mucin is there. And this water, this mucin, the phosphates which are being synthesized by these very important mucous cells, they form a strong mucus covering over the wall of the stomach, and it is because of this mucus which has an important protein known as mucin. Mucin is as if lying there like this is the mucin, and all this mucin has the ability to show to show the utmost resistance against the pepsin and against the hydrochloric acid. Hydrochloric acid can't act over the mucin because the mucin from its the N-terminal and it has most of the coiled N-terminus over which quickly hydrochloric acid or the concentrated format of the hydrochloric acid or this acid can't act effectively, and it is this mucin, this barrier which is providing resistance to the wall of the stomach of a normal person against what? Against the respective digestive enzymes as well as against the respective hydrochloric acid which is otherwise present there. And besides that, this was the second phase. The third phase, that is the intestinal phase, in which we have seen the primary role that is being played by the somatostatin. And besides that, along with the somatostatin, there is influence of the sympathetic nerves which regulate the synthesis of such factors which induce the secretion of your gastric glands. And we have seen that there are other factors like vasoactive polypeptide is there, isn't it? Intestinal peptides are there, some psychological factors are there, decreased in chyme content when chyme leads to get exit from the antral canal, isn't it? All such factors lead to the decreased synthesis of the gastrin or the decreased synthesis of the hydrochloric acid and decreased synthesis of all such enzymes which have been otherwise synthesized by the respective cells. And when all this happens, this all happening ultimately leads to the partial digestion of the proteins, partial digestion to some extent of the lipids, isn't it? And then the respective bolus is not remaining in that condition, then the respective bolus gets converted into another format of the food which is known as chyme. That chyme is just like a paste. Then the chyme prevails there as a paste. After the chyme has been produced up to some extent, then these sympathetic nerves come into play, and then there is least release of some these nervous factors like acetylcholine, which would have been otherwise synthesized by these respective neurons to induce these cells. Then less quantity of acetylcholine like substances are synthesized, and such reflexes come into play, and they induce the myoenteric plexuses which were present in the wall of the stomach to exhibit certain specific movements, like that of the undulatory movements or segmental movements or the peristaltic movements, which ultimately lead to the opening of this very sphincter known as pyloric sphincter. It partially is opened up. When it partially is opened up, then this chyme gets diffused deep, gets transported deep into our duodenum. But such impact is not prevailing over the major or the primary entrance gate known as cardiac sphincter, because as soon as we complete our feeding, our cardiac sphincter is as if closed. Besides that, the lowermost part of our esophagus is as if closed, so that if we will be moving, if we will be drifting after completion of our meals, if we will be jumping, the extra pressure which we will be put over our this cardiac sphincter won't lead to opening of the same and won't lead to the inculcation of this strong acidic gastric juice into our esophagus. So it is necessary for we people that when we are taking food, we should take it after having a proper sitting and after having a proper time, and we should not take food and other eatables while walking, while jumping, while doing exercise, so on and so forth. Otherwise that can lead to, since you know that the swallowing process leads to opening of the cardiac sphincter, when you are doing the exercises at the very same time you are inculcating the food into the stomach, then the extra exercise generating pressure can lead to the outward flow of the gastric secretions into your esophagus, and that can lead to formation of the esophageal ulcerations as well. So with this we will conclude our today's lecture, in which we came to know the process of the digestion in the stomach and the role played by the different cells and some factors in the regulation and initiation of the synthesis of the different products from the parietal cells, or from what? From the peptic cells, or from your chief cells, or from your D or the G cells. Thanks